Starter. Good morning, everyone. I'm Sridevi, Assistant Professor of Mallikarjuna College of Pharmacy. Cordially invite you all to this 5 days International Faculty Development Program organized by Mallikarjuna College of Pharmacy in association with APTI Telangana branch. On the theme, Next Generation Pharmacy Curriculum, Enhancing the Core Subjects with the Digital Tools, AI, and Machine Learning. This FDP has been thoughtfully designed with the objective of supporting faculty members in seamlessly integrating AI and digital tools into core pharmacy subjects, developing the familiarity with the machine learning and virtual laboratories, strengthening the outcome-based education and smart assessment methods. Over the next 5 days, we look forward to engaging in meaningful discussions, interactive sessions, demonstrations, and valuable exchanges of ideas. I would also like to express my sincere gratitude to our esteemed chief guest, Chinala Krishnamohan Garu, who is the President APTI of Telangana branch, and our today's resource person, Dr. P. Mallikarjuna Sir, Professor and Dean of Research at Newcastle University, Malaysia. I also welcome all our participants to this faculty development program. Now, I would like to introduce our principal, Dr. N. Sudhakar Sir, who is the principal and professor of Mallikarjuna College of Pharmacy. He is a distinguished academician and an experienced administrator who has been playing a pivotal role in the growth and development of the institution. He completed his B Pharmacy from Andhra University, M Pharmacy from JNU Hyderabad. He is a specific researcher with the 239 research publications in the national and international journals. The college profile also lists the numerous publications authored or co-authored by him. His contribution to the academic research extends beyond the publication. He successfully guided numerous PhD scholars, 18 awarded under his guidance, and he is currently guiding 6 PhD scholars under Osmania University and JNUH. He is a life member of professional organizations including the IPA, APTI, and IPGA. Now, I request you, sir, to please deliver the opening remarks of a 5 days international FTP program.
Good morning, all.
Unmute, sir.
Good morning, all, my dear pharmacy faculty all over the world. I welcome you all for this 5 days FDP organized by our college and in association with the APTI, Association of Pharmacy Teachers of India, Telangana branch. Respected Mallikarjuna Garu, our chairman, and respected Mallikarjuna Garu is the resource person today. He is from the UK actually. He's the head office in UK, professor and dean. And my co-principal, Dr. Chinnala Krishna Mohan, principal in Hyderabad, and he is the president of the APTI Telangana branch. And about the college started in 2004 with the approval of the AICT and PCI. And later on, in 2008, we started the PharmD program. 2009, we started the MPharmacy program and PharmD PB program also. And pharmacology started in 2010, analysis branch started in 2011. And about the college is having the NBA, National Board of Accreditation, for the last 14 years. It's our 4th cycle is going on. Recently, we have this IIRF ranking also, 2nd rank in Telangana state. And NIRF, for the last 3 years, we're having and expecting a good result very soon. And about the college, the total strength is about some 700 students, and about some 45 students are going out every year for the M-Pharmacy, different specializations and all. And every year, we used to have about some 75 to 100 publications from the college, all in the indexed journals only. We are devoting ourselves for the R&D works in the college. And about the faculty and all, always they are busy with R&D works and authoring some textbooks and all. And about the title of this 5-day session, Next Generation Pharmacy Curriculum, Enhancing the Core Subjects with the Digital Tools, AI, and Machine Learning. With the help of the great dignitaries of the pharmacy profession, about some 10 people are going to help us for the next 5 days. What is the objectives of this FDP? To seamlessly implement the new PCI syllabus by integrating AI and digital tools into the core pharmacy subjects. We all know that from 26 onwards, we are going to have the new syllabus. Lot of changes in the syllabus. Lot number of changes in the syllabus from time to time, particularly 16 is different, 26 is different. The syllabus is devised as per the needs of the pharmacy profession for the next decade and all. So what is there in the syllabus? The new syllabus and all about maybe Python, data analysis, machine learning. So many points are included in this one. So first of all, faculty, as a faculty, we must know what are the things in the new syllabus. We have to upgrade ourselves. We studied our BPharm and MPharm long time back. It's not that one. Every day we have to update. So for that reason, we started this FDP. And even this program trains the faculty in using the machine learning and virtual labs for the modern interactive pedagogy to align the updated curriculum with the OBE, outcome-based education, and smart assessment methods. This FDP to bridge the gap between the industry and academia by fostering the future-ready, tech-driven pharmacy education. Thank you all for this today's session. Once again, my heartful thanks to Arjun Mallikarjuna sir, and even Krishnamohan sir for associating with us for this APTI 5 days special 5 days FDP. Now I request our madam to proceed.
Thank you so much, sir, for your inaugural speech. Now it's giving me an immense pleasure to introduce Dr. Krishna Mohan Chinala, who is the President, APTI of Telangana branch. Sir is a teacher of pharmaceutical sciences, having 25 years of teaching and research experience. He worked as hospital and clinical pharmacist at Kingdom of Saudi Arabia, and he is a psychologist too. Dr. Chinnala, currently working as a professor and dean at a School of Pharmacy, Nalla Narsimha Reddy Education Society Group of Institution, Hyderabad, Telangana. He did his undergraduation of B Pharmacy in Kakatiya University, M.Pharmacy from Andhra University, and PhD from Kakatiya University, and Psychology from KU. He has 115 publications in peer-reviewed journals and 60 presentations. He guided 60 M.Pharm students, and he is a life member of IPA, IPS, APTI, IPGA, LASA, STOX, ISTE, APP, and SPER. He received various awards like APP Distinguished Teacher Award in 2017, Outstanding Scientist Award in 2022, Best Pharmacy Teacher Award in 2022, and Most Socially Active Principal of the Year 2023. Now, I request you, sir, to please tell a few words about today's session.
Yeah, thank you, ma'am. Am I audible?
Sir, you're not audible, sir.
It is unmute only, ma'am. Now, ma'am, please check.
Yes, sir.
Oh, thank you for the confirmation. Respected dignitaries, Sudhakar sir, Principal and Professor, Mallikarjuna College of Pharmacy, and co-convener Dr. S. Sridhar, and other team members of Mallikarjuna College of Pharmacy, and today's resource person Mallikarjuna garu, and my dear fellow teaching fraternity, good morning everyone. It is a great initiation by Mallikarjuna College of Pharmacy to upgrade our faculty, as already Sudhakar sir said. Upgrading ourselves is good, but I can say if you are not upgrading yourself, you will be outdated. So everywhere in whether it is teaching or any job, professionally, if you are not upgrading yourself, you will be outdated, and outdated people are, of course, we are not using outdated mobiles now. Smart mobiles we are using, means upgraded one only everyone will accept. So my request to all the faculty members, please utilize this opportunity to improve yourself. And always I am saying, uh, focus on ask, attitude, skill and knowledge. Of course it must be there with the faculty, and we have to import the same to all our students also. Attitude plays top place, and the next is skill, and next is knowledge. And in the objectives of this faculty development program itself, they have mentioned that to import the knowledge of a new syllabus which is revised by Pharmacy Council of India. And we have to appreciate Dr. Deepender Singh, who is the head for this education division, and he has taken so many uh, pains to implement this syllabus. And this syllabus gets tech upgradation, and it is useful in almost all areas. Like our syllabus for B Farm is industry oriented, and Farm D is clinical research, and M Farm also for research and development. So in all areas, the syllabus is important, the new syllabus. And here Python for pharma applications is there, biostatistics and data analytics introduced. This is very much important in research, and machine learning in how to do new research or reduce the time to discover or a molecule or develop a molecule. New drug discovery and development using machine learning and artificial intelligence in pharmaceutical sciences, in entire healthcare also. Everyone is talking about artificial intelligence in everywhere. And recently I have attended a workshop in Yashoda hospitals, where international speakers came here to Hyderabad and delivered lectures in that workshop. It is like AI in healthcare. So here, of course, we are the team of healthcare, right, along with the doctor and nurse. So we have to update ourselves in artificial intelligence also. Artificial intelligence for natural product research is there. Artificial intelligence assisted drug discovery training is also there in the syllabus. And ADMET prediction studies, this is very much important, the teachers of PharmD, because absorption, distribution, metabolism, excretion and toxicity studies, of course, for BPharm and MPharm faculty also important, but most of the PharmD students are joining clinical research. So as a teacher, we have to be up to date to impart this knowledge to them. And machine learning for formulation optimization, of course, it is important for all the pharmaceutics faculty. And AI in manufacturing and quality control. Manufacturing, pharmaceutics and quality control is for analysis brand. And computer aided drug design for medicinal chemistry, and clinical decision making for pharmacy, and AI for adverse reaction prediction, pharmacovigilance. So nowadays it is becoming popular in India also, pharmacovigilance program, which is started by Indian Pharmacopoeial Commission, Ghaziabad, and they're improving the number of ADR reporting in India. Now we can get the statistics. Previously it was not that good, but of course technology is improved, and this AI may improve further to report all the adverse drug reactions happening in India also. Okay, and dose optimization using AI tools. It is very much important for every pharmacist in dispensing the medicines and patient counseling. Everywhere artificial intelligence is useful. And don't think that artificial intelligence will remove the jobs of any professional or so. It is a additional tool to improve our services. We are in service sector as a faculty or as a pharmacist. We have to think about service to the needy people in the society. So for that, as a teacher, everywhere I am saying Brahma Swarupamude Madhyanet Mahavishnu Sayam Sanjat Shivam Trimurticha Divakaraha. So everyone is there in sun, Surya, the creator, the protector, and the destroyer. In the same manner, a teacher is a creator of good citizen of India and protector of his or her students and destroyer of bad elements of his or her students. So a teacher also should act like a son and should have all these qualities: creator, protector, and destroyer. Of course, as a pharmacist, we are the creators of medicines, we are the protectors of medicines, and we are the one destroying all expired medicines and saving the lives of mankind. So from this faculty development program, already Sudhakar sir said about the artificial intelligence, and of course the faculty of everywhere working with the triple D, discipline, determination, and dedication. But institute is insisting them to work for NAC, NBA, NARF, and in the one of the objectives is for outcome based education. So here the assessment tools and all also included in the objective of this faculty development program. It is a great initiative because everyone must know about NAC, NBA, and NARF rankings and how to work for those things. Then only our institutions will get those ranks. And of course, the third objective is bridge the industry academia gap. This is very much needed now because everyone is commenting on our students that your students are not industry ready. You people are saying, but your students are not industry ready. So with this faculty development program, at least uh, we have to improve ourselves, how to bridge the gap between industry and academia, and how to prepare our students industry ready. I thank the management of Mallikarjuna College of Pharmacy and the principal Sudhakar sir and my fellow teachers for giving me this opportunity for to interact with you all. Thank you. I wish this 5-day faculty development program a grand success. Thank you.
Thank you, sir. Thank you so much for your valuable words. They are truly enriching and have set the tone for this FTP. Thank you once again for joining in this inaugural session of FTP, sir. Now, it is much delighted now to introduce our today's speaker of the session, Dr. Pichka Mallikarjuna, sir. He is a professor and dean of research at Newcastle University, Malaysia. Throughout his career, he has remained committed to advancing knowledge in pharmaceutical sciences. He obtained 600 million USD research grants and he has authored 150 indexed publications. He has secured patents in Malaysia, UK, and Germany. In Malaysia, Sir has played a pivotal role in developing the curricula for pharmacy and other health professions that prepares future-ready graduates. These programs are accredited in Australia, New Zealand, Singapore, USA, and UK. I welcome you, sir, and request you to start the session for today's session, sir.
Thank you, ma'am. Okay. Can you all see my slides, please?
Yes, sir, we can see your slides.
Okay. Thanks, ma'am. Okay. Good morning to everyone. Okay. Today I'm going to talk about the applications of AI, Python, and machine learning in drug discovery and development. Okay. Now I understand that actually the main purpose of this objective, my talk, is what the teaching faculty should teach to the students, okay, in the
Sir, your
low,
volume is very low.
low, madam.
Yes, sir.
Now can you hear me, madam? No.
No, increase the speech. Increase your mics, sir.
Yeah, 1 minute, madam. What about now, madam? Hello.
Okay, okay, sir. Okay.
Now is it okay? Is it okay, madam? No. Hello. Is my voice okay? Hello. Is my voice okay, madam? Hello. 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 Is my voice okay, madam? Someone can reply. Hello, madam.
Sir, it is fine. It's okay, sir.
Okay. Now you can hear me. Am I right? Yeah. Okay. Okay. 1 minute.
So you can continue.
Yeah. Just my slides are not moving. That's why I'm checking actually. Now, yes, my slides are moving, madam.
Yes, sir. Yes, sir.
Okay. Now we look at this drug discovery and development. Okay, and based on my experience, sometimes the students get confused, and also sometimes the faculty also get confused between what is the difference between discovery and development. Actually, discovery and development, they answer two different questions. Okay, drug discovery means it creating a candidate, okay, the candidate that can go into a clinical trial, and mostly this candidate means the molecule, okay, that is developed in a laboratory setting. Okay, it starts with a disease biology and ends with a candidate nomination. Here the candidate nomination means nomination to the clinical trial. So in this phase, what are the decisions that we make is, first we identify what is the enzyme or protein or receptor that we should work on. Then we need to find out a molecule that alters the action of or the biological activity of a particular protein. Then we synthesize a few molecules and we look at their chemical structure, and we also see that what is the influence of the structural changes on the biological activity of those compounds. Then we also look at the selectivity. Selectivity means whether the compound of our interest is hitting the protein that we are interested in and sparing other proteins. Okay, that is called the selectivity. And also we look at the physicochemical properties, okay, to make sure that the molecule, when it goes into a clinical trial, it will have an appropriate absorption, and it will also have a sufficient half-life and also the bioavailability. Okay, we also look at this ADMET properties, that is absorption, distribution, metabolism, excretion, and toxicity. Most of the pharmacology textbooks, what we see is only ADME profile. Okay, and also they talk about the safety together. Of course, that is one way of teaching. The another way of teaching is putting toxicity into this profile. And also the developability. Developability means whatever the molecule that we synthesize in a discovery phase, whether it can be developed into a formulation, okay, that is called as a developability. And also the another meaning of developability is whatever the molecule that we are synthesized in the discovery phase can be synthesized in grams or in kgs. Uncertainty is reduced through design, make, test, analyze, and again design. Actually, we'll call this as a mantra of drug discovery. That means we'll design the molecule first using either computational tools or using artificial intelligence or using a human intelligence. Then we will make that compound. Then we will test it in a biological activity and we will analyze the results whether they are appropriate to go to the next step. If they are not appropriate, again we have to redesign. Okay, that is about a drug discovery. When it comes to a drug development, it is called as establish a medicine. Okay, usually uh, the students or the faculty, we try to use the word drug. Okay, a drug means, in general people terminology, a drug means which is an illicit drug, okay, which can be a ganja or which can be cannabis. Okay, and in that's why in general layman's terms, when we use the word drug, they think that the drug means a heroin or a cocaine or a ganja. Okay, so medicine means, the layman people use think that medicine means which is used for treating a particular disease. Okay, but in a pharmacy profession, we seldom distinguish between a drug and a medicine. Okay, so when we are talking about drug, the people, general lay people may think that we are talking about illicit drugs. So we need to be very careful. And also we need to tell to the students that it is always appropriate to use the word development of new medicines rather than development of new drugs, okay, in a layman terminology. Okay, so it starts with a nominated candidate and builds evidence for the patients and regulators. So the drug development means approval stage and also the clinical trials, in which we talk about the toxicology, whether it is causing any toxic properties in a human being, whether we can have a proper formulation, and CMC, that is chemistry, manufacturing, and controls. Okay, and also first in human dose, it is called as FIH, and the exposure and response, that means how long this molecule is exposed in a biological environment and what is the response. We look at efficacy. Here efficacy means what is the dose that is required or what is the amount of the medicine that is required to produce a desired effect, and the benefit to the risk ratio. We always need to understand that until now there is no single medicine which do not have either side effects or adverse events. That's why if you refer a pharmacology textbook, you always see pharmacological uses, always you see the side effects and see the adverse events. From that, what we understand is it is difficult to develop a medicine without having any side effects or adverse events. Okay, the manufacturing control, this is what we talk about, the quality control or a quality assurance or GMP practices. And the pharmacovigilance is, that's what Professor Krishna explained, when the medicine is into the market. Okay, what different people will react, will respond in a different way. That's why now the people are calling it as a precision medicine or a personalized medicine. In simple words, what does it mean is same drug may not, or same medicine may not work in the same way in all the humans. Okay, that's why the pharmacovigilance is becoming more and more popular. And our PharmD students are mainly are the ones who are expected to work in this pharmacovigilance department in future. What this artificial intelligence do is artificial intelligence supports in all these events, okay, but it do not relax means we can skip any of these steps, we cannot do it, okay, and still we need to do the lab to control. And what this AI can do is it can generate the new ideas much faster, and it can shorten the drug discovery and development cycle to a shorter periods. Usually it is at 12 years, but now with by using artificial intelligence, okay, we can reduce to 3 to 6 years, okay. Then AI is an umbrella and machine learning is one of the methods, and Python is infrastructure. Actually Python, actually if anyone wants to do develop AI or use of AI in drug discovery or machine learning in drug discovery, there is also called deep learning, which is a part of the machine learning, okay. Um, they need to program it, and that program is being done by using this software called Python, okay. So please remember that Python is not an artificial intelligence. It is the orchestration language that connects various databases when it is related to the drug discovery and development, cheminformatics, statistics, machine learning, and also visualization. And whoever learns Python, actually the Python contains a lot of packages or the languages, and all these are the various packages that are there in the Python, okay, which the people can use it, okay. And artificial intelligence, it refers to the systems that perform tasks, okay, associated with a perception. So what we perceive, maybe that means in come to the drug discovery, we may think that, oh, this particular molecule may hit a particular target, therefore it may treat a disease. It will also give you an appropriate reasoning, okay, on what basis it thinks that the particular molecule binds to a particular protein, and also it will predict, okay, how tightly it will bind to a protein, okay, and it also can generate a new ideas. Whereas the machine learning estimates patterns from data. That means when you give some kind of a input, suppose when you put a journal of medicinal chemistry publications into the machine learning, okay, a few papers, and you ask them that, okay, based on these papers, can you generate any new ideas on the probable chemical molecule that can work on that particular protein? Thus this machine learning can do it. And of course deep learning means it's exactly like our brain, okay. It takes the data from different layers, and it works further strengthening this output of this machine learning. Of course the generative AI is the now what the people are talking about, uh, what is this called is ChatGPT, Claude, Claude or Gemini. All these things comes under generative AI. And nowadays the people are using generative AI actually to prepare the lecture notes by the lecturers, okay. The people are using generative AI, the students are using generative AI to find the answers, okay. The administrators are using generative AI to write emails, okay. And researchers are using generative AI to write the publications, okay. And the same generative AI can also be used in drug discovery, okay. These are the various Python libraries, okay. All these things here I have given what is its purpose, okay, and how it can be used in a drug discovery example, okay. This is only a snapshot of the Python libraries that are available in Python, and also which we can use it in drug discovery and development. Of course if I start explaining all these things, it is going to take a lot of time, and my suggestion to the both the lecturers and also to the students is, we are not a computer science persons, we are not, okay. it's very difficult for us to really code the Python language. Then you may ask me, what way then we can teach the Python, or in what way we can make our students to use Python in drug discovery and development. The message here is, we should, that means the pharmacy professionals must work together with the computer scientists, okay, or computer sciences professionals, and we must understand what computer professional is talking about, okay. That means we must speak in a language so that the computer science professional can understand. But if you get it wrong, if we assume that the pharmacy professional should be an experienced person in Python, then it is going to be lead to a disaster, okay, because it's highly impossible, based on my experiences, okay. But when it will be successful means, when computer scientist gives a particular code, you must be able to understand what that code is and what does it mean. That's why here I have given this meaning and purpose. The reason for that is, when computer scientist gives you something, so you need to understand what does it mean and what is its purpose in pharmaceutical sciences, and how we can use that in a drug discovery example, okay. Originally what I thought was, maybe I should talk about Python, but it is going to bore you people, okay. That's why I thought, there is lots of AI platforms that are readily available in open source platform, okay, where we can use it without knowing what Python code they use. Because since these are the open source tools, majority of the Python coding that they use are validated, okay, scientifically validated. So whenever we say that, okay, every useful model begins with a decision. That means uh, nowadays, it's a bit unfortunate to see that, when I met the people, when I asked them that, why are you doing research, the simple answer is, oh, I am doing this research because I want to write a paper, okay. Oh, I am doing this research because no one has developed sustained release formulation for paracetamol. Actually, that is not the right way of doing research. The right way of doing research is, what is the actual problem that the patients are facing, or what is the actual problem that the doctors are facing, or what is the actual problem that industry people are facing in preparing a particular formulation. That is the right question, okay. That's why nowadays, when we ask the people, oh, I am synthesizing the molecules, if you ask, why you are synthesizing molecules, hey Arjun, do you know that when I synthesize this molecule, I can write a paper? For what? Writing a research paper for what? Okay, we really need to understand, okay. And of course, we also know that we want to go for the NIRF ranking, want to go to the NBA ranking, all these things. But if you ask our QS rankings, we need to understand the basic ideology behind these NIRF rankings or NBA rankings or whatever you think, okay, or QS rankings. The ideology behind these rankings is, we are going to produce a competent graduates who can do the research when they graduate, and that research is going to help the humankind. That is the main thing. But if you simply saying this one, oh, my NRF ranking increases if I write a publications. Yes, simply write publications without having any end use, okay. Okay, having a patents is good for me. Okay, do something patent for the sake of doing a patent. Okay, so that is not the main purpose of these rankings. Okay, the main purpose of the rankings is to produce the competent graduates, okay, which are coming from the institution, okay, which is highly ranked in NIRF. That means they are doing the best research, and that best research is helping the students, okay, to gain that knowledge, and after graduation they use their research, okay, to do something good to the humankind. Okay, that's why we need to know what is the decision, what compound you want to synthesize, what patient, okay, where it is, what is the batch decision, okay, and endpoint, what are the measurable outcome represent success. Okay, at this point of time we are looking at NIRF ranking and all those things as a success, but in future you will see that, okay, is this highly ranked NRIF institution, what kind of a innovation that they did that helps the mankind in terms of, instead of how many papers they publish, okay, or how many patents they have. Okay, then these data are examples relevant, traceable, and representative. Okay, and of course for everything there is a model and there is a validation. And the people who are teaching medicinal chemistry, they already know that there is a docking. Okay, there is also QSAR or structure activity relationship. Actually all those are AI models. Okay, AI is not new. Okay, sometimes we think that, oh, AI is a new thing in your drug discovery and development. It's not. All the computational tools that the people use, that we have been studying for the past 10 years, okay, like a QSR, all those are AI models. Okay, but the word AI is new to the people nowadays. Okay, do not begin with which AI model should we use, and we always begin with which decision is uncertain, costly, or slow. That means which decision, that means why the patients are not responding to a particular medicine. That should be a starting point for you to think why this particular medicine, let us say now in anti-diabetic drugs, okay, there are new medicines came, they are very expensive, so you need to do a research, why is it so expensive, what can I do to reduce its cost. Okay, or slow means in the drug discovery, why I should use antibiotics 5 days, 2 times a day, at least for 5 days, can I reduce it. Okay, or why I have to take a paracetamol tablet 3 times a day, why can't I take 1 time a day. Okay, that is called sustainability. So these are the questions that we really need to ask. Okay, then actually I thought that what should I talk actually in this one. Then I thought that I'll take one example, like a non-small cell lung cancer, okay, in which I will take a particular molecule, okay, a particular target, that is EGFR. Then basing on that one, I thought of explaining the things, because we need to understand, when we buy a phone, okay, or when we buy something, it comes with some kind of a booklet, or nowadays they will ask us to look at the internet, go to the particular website and read. Actually, to be honest, none of us who are sitting here, we never read that manual at all. When we bought this one, we'll try to start, and we'll try to what is this called, we'll try to switch it on, and we'll try to use it. Whenever we have a problem, then only we go and see the someone, hey, I'm not getting this problem, I'm not finding this option. So whether we can speak to our friends, or we speak to the, if it is a problem is big, we speak to the person who sold that one. Okay, so this is having a key message. Once upon a time, okay, all we thought was we should know the theory first, then only we should apply. But just now I have given one example, you just to reflect on it. Or do you have really the theory behind this particular phone that you are using? No, you are trying to use it immediately. Okay, now the present generation Gen G students are also like that. Okay, of course, actually I do not know that, actually I did not realize that this particular FDP is to train the students. If I know that, I might have prepared the presentation in a different way, what Gen G expects. Okay, so that's why in a similar way, we also need to understand that, okay, when I studied, I do not have a textbooks, I refer the senior notes. Then afterwards the textbooks came, afterwards the guides came, afterwards Google came, and afterwards the chat GPT came. So whatever we teach to the students, I think the present generation students, they don't even listen what we say, because they think that ChatGPT is still the better, Gemini is still the better, Claude is the still better, or Google is also the better. And surprisingly, sometimes I am seeing the lecturers also nowadays, they're not even referring the textbooks. They are simply searching the Google and they are preparing the lecture notes. That is the truth. Okay. And there is nothing wrong with it. But what I'm saying is we need to go with the flow. Okay. So that's why I thought of maybe taking an example, then explain it so that you can understand in a better way. Okay, so the decision, okay, 10,000 ideas, then because if you want to spend all these 10,000 ideas into the lab, it is going to take a lot of time, it is going to take a lot of money. But out of 10,000 ideas, I think you can reduce to the 20. Okay, then this is the decision that you have to make at the beginning. That means how many molecules I am going to test in the lab. Okay, and the endpoint means what exactly you want to do, or whatever the molecule that I'm going to do is, it is going to act on this EGFR, epidermal growth factor receptor, and whether my compound is going to be soluble or not. Okay, whether it does not have any toxicity profile. Okay, then how you can get this one is you need to go back to the data. Okay, that is reported in the literature, that is called as the literature search. Okay, then you look at all the molecules and you look at the real lab results. Okay, during my PhD, my literature search itself will take about 6 months to 1 year. Even when I do that literature search also, I'm not sure that whether it is complete or not. But now with the use of AI, I can complete that literature search and read everything within a month. Okay, of course, nowadays the people are doing it in 1 day, or some people are doing it in 1 hour. But I can say that yes, they are doing it, but they are not checking. That's one of the problems. Okay, then you need to look at the models. Okay, previously what I used to do is I need to draw all the structures on a piece of paper, then I need to hold my head and I need to check what is there, what is the similarity, that is structure activity relationship. Now AI can make it much faster. Then you need to really make it, that means you synthesize it and you test it, and maybe you can with 4 hits with AI versus one usually. Okay, that is the thing. Okay, that means when you do now with your human brain, you may get only one compound, but with AI you may get this one. Okay, new laboratory results become data for the next round. Again, this one will go into this will become the data and it will go like this. Okay, and this is the summary of the traditional discovery versus AI assisted discovery. Always remember that we are making use of AI as our PhD student. Okay, you should not depend on the AI. But nowadays the people say that, oh, AI is doing everything for me. Okay, that's why they blindly believe whatever they say, it's not right. Okay, traditional flow, it is a literature driven small library screening. We synthesize only few molecules, and we don't look at the ADMET properties at the beginning, and we do the experimental validation. Whereas in the case of AI-assisted workflow, actually you can do target prioritization. That means which protein that you should work on. You can look at the millions of compounds. Okay, then you can use the generative AI to get the new ideas, and you can do multi-parameter optimization. That means you can do the activity, you can do the ADME profile, and also you can do the experimental validation. Okay. AI accelerates the cycle but does not remove the need for biochemical, cellular, pharmacokinetic, and clinical validation. Because I want to emphasize this one. Suppose we being a teachers, if we do not tell this to the students may think that AI is the everything. Okay. And if that is the one, okay, and just here, because it's not the purpose of this lecture, but I want to highlight one thing. Nowadays, recently the MIT did a research. The people who are going to use AI, they are going to get dementia, they are going to get Alzheimer's disease, they are going to get a Parkinson's disease. The people are expecting that the people are going to get a new neurodegenerative diseases. The reason for that is, since there is an AI, the people started not using the brain. When the people do not use the brain do not work. When the brain do not work properly, if it is not sharp, the dementia, forgetfulness, Parkinson's disease, Alzheimer's disease, this is what we are going to see after 2035. It's guaranteed. It's really guaranteed that. Because a simple example is, you buy a knife, okay, you store it. If you don't use it, after some time, it will get rusted. In a similar way, the brain is here. If you don't use it, simply use generative AI, then it will get rusted. That's the thing of AI. Of course, now the people are talking, oh, AI is so great, but the people are not realizing that what is the problem that the future generation is going to face. And in general, Gen Z, yes, they are going to have lots of issues with neurodegenerative diseases. Okay. Always start with the end use. This is called as a target product profile, which is simply called as a TPP, which is being used in any drug discovery company. Okay. Whenever they want to start a new project, they call it as what is TPP. Okay. So we need to decide which patient that you want to targeting. Okay. Of course, here all these are the mutations. Okay, mutations means a particular amino acid in a particular gene will be mutated to another amino acid. So because of the change in the amino acid, it will cause a disease. Okay. Then desired efficacy, then what you want is whether you want to target it at a 10 milligrams or 100 milligrams or 500 milligrams. Okay. And selectivity, a prefer mutant EGFR over wild type EGFR. Wild type EGFR means it's a normal EGFR which is there in all the humans. What we want is, we want to work only on the mutant EGFR, not on the wild type EGFR. And developability, that means whether it is having a proper appropriate solubility, whether it is stable in the liver, whether we can it does not have any drug interaction receive, whether it has appropriate bioavailability. Of course, the clinical setting, okay, whether you have to decide whether it is going to be a first line drug or second line drug or this one, or whether it is going to develop any resistance. Of course, here we are talking about NSCLC means non-small cell lung cancer. Okay, but even though this lung cancer, what will happen means cancer will spread to the other organs, which is called as a metastasis. Okay, so the people who has a non-small cell lung cancer, they also spread the cancer to the central nervous system. Okay, and these are the various things about the one about the protein. Now, data foundation for AI assisted drug discovery. For if you want to use AI, there should be a data. Without data, you cannot do anything. Okay. Suppose if you want to know the data about a particular protein, these are the websites that you need to look at: GeneCards, PubMed, and String. Previously, I used to read all these the information that is present in the GeneCards, but when the AI comes, actually I can make use of AI to summarize for me this data. Otherwise, it is going to take a long time. Then the structure, whenever I refer the structure, please don't get confused. This is not the structure of the chemical compound. Here, the structure means the structure of the protein. In drug discovery, whenever we use a word structure, that means it is referring to the chemical structure of the protein or receptor or uh, enzyme. Okay, so these are the two websites, AlphaFold and RCSB PDB, where you can find the three-dimensional structure of the proteins. Then the known ligands. Ligand means the compound, chemical compound that is binding to a particular enzyme. So it is ChemBel and PubChem. Okay. Then developability. Then these are the tools. Actually, behind all these tools, am I right? The Python is included. Python is there. So Python is the backdrop. Okay. Now, see, this is simple thing about the EGFR structure and function. Okay, this is the epidermal growth factor receptor. Okay, what will happen means when the EGF or TGF alpha they bind, okay, they produce pathways. Okay, and these are the proliferation and differentiation, survival and growth, and inflammation and immune signal. Actually, when someone wants to develop a research proposal or when someone is teaching about a drug discovery, actually without these kind of diagrams, we cannot talk about anything about drug discovery and development because it's very difficult for the people to understand. Okay, in normal cells is controlled. That means when the TGF alpha ligand binds to this one, then only all these thing pathways will propagate. Okay, whereas in the and when there is no TGF alpha, then there is no proliferation. Okay, here you can see the proliferation means cancer cells will grow much faster. Okay, differentiation means normal cell will be transformed into a cancer cell. Survival means whenever we give a medicine, the cancer cell will try to survive. Growth means the cells will try to grow much faster pace. Okay, then because of this one, it causes some kind of a inflammation that relates to the pain, cancer pain, and also our immunity will go down. So once the person gets a cancer, they will also get lots of other diseases. Okay, so in a normal EGFR, it goes like this, whereas in the case of a small cell lung cancer, all this will be continuously will go. That means even though there is no this particular one, it will continuously go. Okay. Now, the another important thing is in drug discovery, it's only 20 amino acids. That's all the proteins, receptors, enzymes are made up of only these 20 amino acids. If you know what are they, if you know what is their chemistry, then you can easily understand. And what happens is when we teach to the students, we same thing we teach like a 1 hour lecture, so or 2 hours lecture, and the people cannot grasp it. So the way that we should teach is in a single slide. If we can put them in a way that by looking at this slide, they can understand what is the full name, what is the 3-letter code, what is the 1-letter code, what is its nature, okay, whether it is a hydrogen bond donor or whether it is a hydrogen bond acceptor or whether it is a non-polar or a polar or it is aromatic or whether it is acidic or basic or neutral. So this is how we should teach to the students, Gen Z students, because they want to get everything in a single slide. Okay, of course, if you look at my previous slide also, in single slide I'm talking about everything. Okay, then actually suppose if the student do not understand this, actually the same slide they can put it into the chat GPT and it can ask, they can ask the chat GPT to explain it. Actually from this slide itself, chat GPT can give a 20-page notes. Okay, but if the same 20 pages, if we are talking to the students, they may not pay attention. Okay, that's the thing. And here also you can see that EGFR mutations in a non-small lung cancer. So everything is there. Why do EGFR mutations matter? Okay, most clinically important mutations occur in the tyrosine kinase domain. Okay, the increased kinase activity stabilize the active receptor state. The result is continuous cell growth signaling and mutation testing guide selection easier for targeted therapy. In general, what happens means in a traditional lecture, this itself we talk about 1 hour lecture. Okay, that means we try to give more and more information to them. Actually, it's not necessary. You can tell them in a few sentences, and the student who is really interested, they will go and check. Because even though we teach, what happens means if you teach the same thing in 1 hour lecture, what happens means the student lose interest and the student cannot get anything, and at the end the student will go to the generative AI. Instead of that, if you tell them this one and if you ask them to prepare a notes, actually they can prepare a much better notes. Okay, then these are the common because I'm telling this one because just now only I understand that the main purpose of this FDP is how effectively we should teach to the students outcome based curriculum. Okay, and mainly we are teaching to the Gen Z. Okay, so these are the various mutations and these are the various amino acids. Okay, and this is the frequency, this is the effect. Okay, and this is the therapeutic opportunity. Okay, and this is the from mutation to disease, how it goes. Okay, and actually to be honest, actually the people can use AI to prepare these kind of slides. Yeah, and 3 ways AI can help discover candidate molecules. Yes, of course, this is what we say in any medicinal chemistry textbook: ligand based discovery, structure based discovery. Of course, these two we talk. Actually, now this is the one, generative design. Okay, generative design means how we can use ChatGPT in the drug discovery, how we can use Gemini in drug discovery, how we can use cloud in a drug discovery. And of course, predicted activity, selectivity, ADMET, synthesis feasibility, experimental validation plan, everything we can do it. Okay. Now, if you look at again, this is one opportunity where I'm talking about all the chemistry of the entire EGFR inhibitors in a single slide. Okay, what is the first generation, what is the second generation, what is the third generation. Okay, and we are giving their chemical structures. Okay, so of course when I studied, I used to draw the, what is it called, my teacher used to draw the structures on a blackboard. Okay, and usually it will take about 3 to 4 minutes. By the 3 to 4 minutes the student goes to sleep. Okay, that's why we also should use the advanced tools. It's not called as an advanced tool. Okay, we also need to use the chemistry drawing tools to tell to the students so that they can easily see the entire structure without losing their this one. Okay, because by this time you know that Gen Z. Okay, again I'm telling another reason is people used to do the Facebook. Of course, nowadays the most popular thing is Instagram. If you look at the Instagram, okay, if you look at the videos, all the videos are very short videos. Some videos are 5 minutes, some videos are 3 minutes, some videos are 1 minute. Actually, the videos which are 1 minute, they are getting lots of likes. That is Gen Z. That means Gen Z cannot pay attention for more than 30 seconds or more than 1 minute on a particular thing. They need to go. They need to really slide. Okay. This is the structure activity relationship of the EGFR inhibitors. Okay, here also you can see that this same slide also we can put it, no need to prepare in such a nicer way. Okay, we can prepare it in a simple black and white text. When you put in a black and white text, the students lose interest in paying attention what you are talking about. Okay, this is the gefitinib and erlotinib, efatinib and astanib, and here we are looking at the key structure activity relationship features. But suppose if you see any standard medicinal chemistry textbook, the structure activity relationship, they will list down about 10 points or 12 points, and most of the time they are repetitive. Okay, so this is erlotinib, efatinib, and this is key structure activity. And if the people should know what is the difference between these four. Okay, this is the gefitinib, the first ATP competitive EGFR inhibitor. Okay, but it is less potent. That means it requires more amount to produce the desired response, whereas this one is more potent. Okay, and you can see that when this is changed to this moiety, then it became more potent. And afatinib, so what it happens is it is covalently binding to the um, protein. Okay, in general, in medicinal chemistry textbooks, what we say is we always want reversible binding with the protein. We don't want irreversible binding. So we always think that covalent bonding is not good, but actually it's not true. It's not really not true nowadays. Okay. Now this is the mutant selective. So here what happens is all these molecules, they work only on the lung cancer, only at the lung, whereas this one, it can also work on the brain. So that's why this is very advanced one. Okay. Now here what we need to see is, that's why I told you that we need to understand what is the clinical problem, what is the exact problem that the patients are facing, or what is the exact problem the clinicians are facing. Okay. Brain metastases are common in advanced non-small lung cancer and are more frequent in EGFR mutant disease. Okay, that means when the person is diagnosed with cancer, already the cancer was spread to the brain in the 10 to 25% of the patients. Okay, that is too late. And 40 to 50%, so during the treatment period, and 40 to 50% actually will have a brain cancer. Okay, so clinical need is we need to protect the brain and treat existing CNS lesions. That means we need to treat the lung cancer. We also need to treat the brain cancer that is spread. Okay, so why earlier TKI struggles? TKI means tyrosine kinase inhibitor struggle, because they cannot cross the blood-brain barrier. Since they cannot cross the blood-brain barrier, what happens is the drugs that the people use cannot treat the cancer that is spread to the brain, so the people will die. But you see, in medicinal chemistry, what we say that, oh, when we synthesize a molecule, it should not cross the blood-brain barrier. That's what most of the medicinal chemistry textbooks says, and that's what most of the lecturers also teach to the students. But what we need to know here is all the principles that we study in medicinal chemistry is not a hard and thumb rule. Okay, it depends upon the context. Okay, and what is the advantage they got is because that's why the research was happened like this. That's why they could able to get CNS active therapy. Okay, now the other thing is whenever we synthesize the molecule, we use the word drug likeness. Okay, many active molecules, okay, little developability known. That means in a lab we can, okay, let's say, that's why if you see a journal of medicinal chemistry, or something, even any MPharm student or any PhD student do a chemistry research work, they say, oh, I found out the best molecule. Okay, this is very potent. When you look at the conclusions, this is what they say. But unfortunately, no one has asked why the industry is not taking up that molecule. Why those molecules, whatever we synthesize, we think so great, so great, so great, why they are hidden in the PhD thesis, why they are hidden in what is this called as in the papers, research papers, why no industry is taking up, why no doctor is using it, why it is not there in the clinical practice. Okay, that's why we need to know that this is the academic research, and also there is the industry research. When we do the academic research very good, then industry will take up that. Okay, so that's why the drug likeness is very, very important. Okay, that means it should have an appropriate ADMET properties, and it is also have a proper oral bioavailability. Now, these are the some of the physical properties. You see, there are few things: 1, 2, 3, 4, 5, 6, only 6. Okay, but it's very difficult for our brain to look at all these 6 at a time and make a decision. It's very difficult for us. Whereas you can make use of AI to tell that, okay, by looking at all these properties, can you pick me a molecule which is having an appropriate of all these properties? Then it can pick. That is the advantage of the AI. Okay. Of course, we have the Lipinski rule of 5. We can also see the Weber rule. We can also see the other filters. Okay. But most of the time, what we teach is Lipinski rule of 5 is the thing. We emphasize so much when we teach. But have we ever asked why the antibiotics are so big molecules? Why the antibiotics are so big molecules? They do not obey the Lipinski rule of 5, but however, they're being used. Okay. So this is the question that we really need to ask. Okay. So, but when we teach in such a way that Lipinski rule of 5, Lipinski rule of 5, Lipinski rule of 5. So the student think that, yes, if it is more than 500, it's a waste. But when he goes to a practice industry, it has proven to be wrong, because directly that industry person will ask, if you take a kanamycin, what is the molecular weight of the kanamycin? Okay. Or erythromycin, what is the molecular weight? So these are the things that we need to really pay attention when we teach to the students. Okay. That's why Prof. Krishna also say that there is a gap between graduates and the industry needs, and sometimes it is also because how we teach and what we teach. Okay. So use rules to flag risk, not to reject automatically. You can compare the compounds, you can interpret the rules. Okay. And many approved oncology drugs, that is anti-cancer drugs, and of course for also this one, what is this called, antibiotics is also exception. Okay. Rules help prioritize molecules, but medicinal chemistry requires context and judgment. Okay. That is very important. Okay. Then of course, if you want to develop an EGFR inhibitor, what we say that is it should have the sufficient GI absorption. Okay. Since we are taking it as a orally, okay, it should protect itself from the liver enzymes, and it should have a good selectivity over mutant, and it should have a half-life of about 8 hours. Okay. But here when we say that it should not cross the blood-brain barrier, but whereas when you want to do the brain metastasis, then CNS penetration is very important. Okay. Of course, in the brain there is called as a P glycoprotein. Okay. Because our cells also have a particular protein, P glycoprotein, it will try to kick out the molecules. Okay. Then if you are talking about only local lung tumors, this BBB is not required. So this is one of the examples. Then these are the common mistakes. What the people try to do is, okay, simply they say, oh, Lipinski rule of 5, pass or fail. Okay, simply they do it, and they look at only the potency. Oh, my compound is active at nanograms. Okay, but if it is not soluble, what is the use? No use. And sometimes the people use wrong molecular forms. Okay, some the people use a salt form or tautomeric form, which is not the actual thing. Okay, and some people, they do not know that whether, okay, I synthesize the molecule, whether I'm going to give it orally or whether I'm going to give it intravenous or whether I'm going to give it transdermal. People don't know. So sometimes the people over trust the AI and also they ignore the selectivity. Okay. Now, the other thing is Campbell is one software where you can see lots of chemistry associated with bioactivity. Okay, which is much better than any of the medicinal chemistry textbooks. Okay, you will find everything. Okay, so here you can talk about a particular molecule, on which protein it works, and what are the molecules that are being synthesized or acting on that particular target, what is their activity, what is their structure activity relationship, what is their whether we can select the what candidate can be selected, or we can also check the ADMET properties. So what does it mean? Means it is not necessary for those people who are interested in drug discovery, no need to check the research papers. Directly they can go to the Campbell. In the Campbell they can get all the information when we are looking at ligand based drug discovery. Okay. So these are the various data that is available. Okay, what is the compound, what is the chemical structure, okay, what protein it works on, and even you can get the biological assay system also. Okay, and document. You also can get research papers, and also you can also get the mechanism. Okay, and these are the some of the things that you can do it. Okay, just I'm going through so that the take home message is Campbell is the software, open source, you can access it. Okay, where you will get lots of information. That is the take home message. Okay, and you can also see the different ways of expressing the activity. You can look at all those things in the Campbell database. Okay, of course, you also can look at, you can draw the chemical structure, and you can also ask Campbell to find out this thing. Actually, this is called as an AI tool, okay, for which you need not to do, what is this called? You need not to know coding. Without coding also you can do it. Okay, and these are the practical Campbell workflow. Okay, these are the common mistakes. Okay, copying the best IC50 only, you cannot do it. There are sometimes the people ignore the units. Okay, some people, they do not look at what organism it was there, and confusing salts. Okay, and comparing unlike SS. Okay, and for each and everything, what you need to know is AI tools. Of course, Campbell, I can say that is an AI tool. It has both strengths and weaknesses. Okay, you should know what is the strength, and you also should know what is the weakness. Okay, then the other thing is AlphaFold, which is gaining very popular, because if anyone wants to do a structure-based drug design, they need to know the 3D protein structure. Okay, and now here you can get this AlphaFold. Okay, AlphaFold, it can give you the 3D structures. Okay, and most of the time what happens is in medicinal chemistry, we talk about structure-based drug discovery, but we never show them all these websites. So how the people can understand what we are talking about? Okay, if we can show them, actually they understand actually what the lecturer is talking about. Okay. Then this is the AlphaFold protein structural database. What it will do is it will predict 3D structures of the proteins, because we know that in drug discovery, okay, if you want to design a molecule, you need to know the 3D structure of the protein. Okay, so when you know the 3D structure of the protein, then you can design the molecule much better. Okay, and unfortunately, of course, I have seen this revised curriculum, new curriculum. Okay, I know they have integrated few things, but I see that, and they call it as an outcome-based education, but I'm really not sure that how the students can really meet the outputs, okay, or outcomes. Okay, and this is the best way to search the AlphaFold TB. Okay, and for which, because even though I have given you some search workflow, actually if you go to AlphaFold DB, okay, you can get it, or you can go to YouTube, you simply type AlphaFold TB DB videos, lots of videos will come. Okay, in the same way, Campbell also, if you type, you will get a lots of videos, you will get it. This is where Gen Z nowadays, are losing interest in attending the classes, because in YouTube they get a lots of advanced information than what we teach in a classroom. Okay, so what is the best way that what we should do? We should tell to the students that what videos they must watch. That means, because all videos are not good, some videos they give a nonsense information. So as a lecturer, we need to tell them that, hey, you look at these videos, because you just imagine, even though when I'm giving a lecture now, I'm not sure how many of you are really awake. Okay, it's true, it's difficult for us to be awake, but when you go to a movie, imagine even Gen Z also can watch the movie for 2.5 hours. You need to understand what is the difference between when the same Gen Z student come to my class, he cannot pay attention even for half an hour. How come the same Gen Z go to the cinema movie, can watch 2.5 hours, and in the 2.5 hours after come back, that Gen Z student can narrate the entire movie. No need of by hearting, no need of memorizing, and they can memorize much longer. Okay, if you see then of course if you analyze, you know, because when we give a lecture, it's a monotonous. When we see the movie, then there's a lot of things are there, so we need to see how we can do it. Okay, and these are the understanding the AlphaFold entry page. Okay, protein identity, sequence, confidence colors. Okay, you can also download it. And these are the confidence scores that you will get, because all these are the predicted structures. When the confidence score is high, that means its prediction is very high, very good. Okay. Then how we can use AlphaFold models in drug discovery, then for all these things. Hypothesis generation means generating the research idea. Okay. And these are the common mistakes when we use AlphaFold DB. Okay, using the wrong protein, ignoring the confidence scores, and assuming drug binding is shown. Okay, and of course it's too technical, that's why I'm not going into the details. Okay, now there is another website which is called as RCSB PDB. Okay, actually to be when I studied BPharm also, the people talked to me about computer-aided drug design. Okay, MPharm also I was learned. In PhD also I was learned. Later on I was taught to the students. Even now also the faculty are teaching, but the faculty are teaching medicinal chemistry without showing these websites at all. Okay, so then you can see where the gap is. Okay, where the gap is between the industry expectations and what is happening in the academic setup. Okay, and if you want to see the RCSB PDB versus AlphaFold is RCSB, all these are the experimental structures, whereas AlphaFold is only the computational predictions. Okay, both have advantages and disadvantages. Okay, and AlphaFold inventor, okay, he is the 2026 Nobel laureate. Okay, this is the first time in the history of Nobel Prize a person who only through the predictions got the Nobel laureate. Okay, and that shows the AI is really progressing, because some of us who are sitting here, they may think that, oh, AI is a bullshit. Some people may think that AI is very good. But what I can say is AI is getting better and better and better, and it is going to be much better than the human brain. Okay, but the problem is the day will come where the human brains do not know how to use AI. That is the biggest problem. Okay, of course, if you look at the EGFR kinase EGFR as an example, and these are the various steps that the people should use, because when we teach like this, it's easy for the students to understand what we are talking about, rather than simply teaching what is there in the medicinal chemistry textbook. Okay, of course there are the advanced search tips and how to select the right PDB entry. These are the few things, and whatever I'm saying, all these things are available in the YouTube videos, or if you go to this RCSB PDB, there are tutorials available. Okay, and resolution and validation. Okay, because in PDB, when you type the EGFR, you will get lots of structures, but you need to select one of the best structure. Okay, and what are the things that we need to look at it. And this EGFR in the RCSB PDB also, actually you can see what is the interaction between this protein and the molecule. Okay, actually here what happens is here we need to use our brain, okay, whether we want hydrogen bond or hydrophobic or halogen bond or a covalent bond. It depends upon the situation. In some situations, the covalent bond is not important, but in some situations, it is important, okay. Then these are the common mistakes when using RCSB-PDB, okay. Let us say, if you see, whenever I'm teaching, whenever I'm delivering a talk, I'm talking about what that particular software can do or what that particular AI tool can do, okay. What is the best practice to do it? What is its pros and what is its cons? And what is the benefits, advantages, and what is the disadvantages? And what are the mistakes that we do? This is how we should teach to the students, okay. The student should know actually what information they can find, how to search for it, okay, what is the advantage of it, what is the disadvantage of it. Because if they know the disadvantage, they know that, oh, this is where I have to be careful. And also we need to tell them what are the mistakes that they do, what are the common mistakes that can happen, okay. So of course, with AlphaFold, PDB, and everything, if you put this one, AlphaFold, PDB, Campbell, Swiss similarity, and Swiss ADME, this is entire thing is nothing but the medicinal chemistry, okay. Unfortunately, still in medicinal chemistry, we'll ask the students synthesize a particular molecule, give all the steps of the synthesis. If we ask why the really students need to know all the chemical steps now, because if you ask ChatGPT can give you the entire steps, including the synthesis of morphine. When we studied, yes, it's true. At the time there was no ChatGPT, so that's why we need to write. But now the already ChatGPT is there. It can give you whatever the molecule that you give. If you ask them to give the synthetic method, it will give you the synthetic method. But what we need to know is we need to teach to the student whether the ChatGPT is giving the right synthetic process or not, okay, and why it has given wrong, where it has, how it can be corrected. So if you can teach to the students, they do much better, and they will be much more applicable in industry, okay. Even after my B.Pharm or PhD, when I go for interviews, usually the people used to ask me to draw the structures, but I don't think nowadays in the interviews the people are not asking to draw the structures. The people are not asking to draw the entire chemical synthesis. But what the people are asking is, if you are using HCl, what precautions you should take. These are the questions that the people are asking in the interview. Unfortunately, what happens is when we teach chemistry, we don't teach them how to appropriately handle hydrochloric acid. Instead, we see that, oh, whether you got product or not. Okay, these are the things. Now the generative AI, this is a new thing. Okay, what it can do is it can summarize the papers. It can summarize. It can give you a particular EGFR. It can summarize for you. It can also explain the structure activity relationship. But what it cannot give you is it cannot identify the research papers. Okay, sometimes it gives the incorrect IDs. Sometimes it gives the hallucination. Okay, use AI for speed. Use databases for truth. Use experts for decisions. Here, teachers, we are experts. Okay, we are experts. So we have to tell to the students how to use AI effectively, ethically, okay, and also in scientifically and responsibly. And because if teachers think that my job is only content delivery, the problem is generative AI is delivering much better than what we can deliver. Okay, so target rationale, literature trials, molecule ideas, ADMET thinking, clinical context reports, all these things the generative AI can do it, and they can do it effectively and efficiently. Okay, And generative AI is not a single tool. Okay, we have a large language models like these things. We have a search grounded AI like these things. Okay, literature AI, these things, and source grounded notebooks. These are the things. And of course, if you want to design the new molecules, these are the things are there. Okay, this is generative AI tools. Okay, what generative AI can do and cannot do is, okay, yes, that means it will helps you. It helps you in summarizing the information. Okay, it summarizes the table, summaries, everything, whatever you want, depending upon how effectively you use it. Okay, translate technical content, help compare. Even I tell you, suppose if there is a student who is not so good in English, okay, and attended a lecture, when we teach only in English, am I right? But we seldom teach in speak in Telugu. Actually, if the student is smart, he can record our voice. Am I right? He can ask ChatGPT, convert it into Telugu. It will convert. That's why what I'm saying is, nowadays generative AI can do much better than our teachers in the content delivery. Okay, and it's not safe without verification, so we need to verify it. Okay, and these are the pros and cons, okay, of each and every ChatGPT, Claude, Gemini, Copilot. Unfortunately, what happens is ChatGPT and Claude, all these people are using not for scientific purposes. Okay, and most of the time, actually, last time when I visited India, I asked the students in one of the colleges in Yeluru. I asked them, I asked the students how you get the answers. I say, I use ChatGPT. How do you know whether it is correct or not? Oh, I also ask Gemini. If ChatGPT and Gemini both are saying the same thing, actually, then I think that it is right. I'm really surprised to see that they're going to consult the teachers. Okay, so it's a B Farm student. B Farm students are saying, actually, it's the same all over the world. Okay, and whereas in our institutions, we always tell to the students to use AI, and we also tell them where it can go wrong. Okay, and these are the literature AI tools, what the people use in writing a research papers. Okay, and these are the molecule generative AI tools. By using these tools, actually, you can what is that called as you can do new molecules. You can synthesize, you can design the new molecules. Okay, and these are the common mistakes that the people do: asking broad questions, trusting citations, accepting numerical values, ignoring confidentiality, no record of prompts, because the people don't write what is the prompt that they use, and using one AI tool only. So these are the common mistakes that they do. And of course, this is what we do in our standard medicine chemistry textbook, QSR, docking, ADMET model and generative models is not there in the textbooks. Okay, I think I saw even the recent syllabus, but in that one also it is not there. And here, when you want to do the candidate selection, you need to look at all these points: 1, 2, 3, 4, 5, 6, 7, or 8. So what happens is it's very difficult for the human brain to think all these things in a single go. That's why we take AI. Okay, and from AI hit to the lead hand, so AI will give you many candidates. Then you look at the chemicals, any reactive compounds you remove, any duplicates things you remove, any compound that is not synthetically feasible you remove it. Then you look at the structure check, okay, whether the poses are correct or not. Then you look at this ADMET filter, and then you do the experimental essays. Then this is lead hypothesis. Okay, so in a medicinal chemistry textbook you will see a funnel, but that is a very old funnel. In the AI, this is the funnel that we need to look at it. Okay, And this is the early guidance for early career researchers. Start with a patient problem. Don't start with writing a research paper. Then it completely defeats the whole purpose. And build the evidence chain. Then avoid common traps. Okay. So do not start with a tool. Start with a clinical and structural problem. Okay. Then common mistakes in AI-assisted candidate discovery is simply the people go to ChatGPT, Cloud, anything without understanding the strengths and weaknesses of it, and treating the AI output as a fact. That means whatever the AI says that we think that it is good, and we don't look at the selectivity and overvaluing one score. Okay, and the molecule can give anything, but unfortunately nowadays the AI cannot synthesize the molecule. Okay, still we need to synthesize the molecule. Okay, that is one thing. Okay, and AI cannot do the titration. Still we have to do the titration. Okay, so what does it mean is in the AI era, content is not important, skill is the most important. Okay, skill is the most important and interpretation is the most important, and also the documentation. Okay, that is also very important point. Okay, documentation we need to write. Of course AI can help you to draft it. Okay, and of course it doesn't have the ethics. Okay, thank you. I hope I covered everything in 1.5 hour time. Are there any questions? Hello, are you all there? Any questions?
So it's time for questionnaire from the audience. Any questions, sir?
Hello, are you all there?
Yes, sir. Yes.
Is it audible?
Hope everyone is audible. Sir, we have a question from Dr. Sumit Rathod. Please guide us regarding the type of questionnaires and routines using net pharmacology.
What, madam? Net pharmacology.
Yes, sir. Yes.
Okay, the net
Hope everyone is
pharmacology.
audible.
Yeah, net pharmacology actually the people use for natural products mainly, because
Sir, we have a question from Dr. Sumit Rathod.
yeah,
He's asking to guide us regarding the target selection using the network pharmacology.
oh, target selection using network pharmacology. Okay, once upon a time the network pharmacology was
Sir, we can't
considered.
hear you.
You can't hear me. I'm speaking. Hello, I'm speaking. Hello, hello. Hello, madam. Can you hear me now? Hello, hello, hello. Can you hear me or no?
Sir, you're not audible, sir.
Now is it audible? I don't know why it is not audible because I'm speaking.
Yeah, sir, it's audible.
Okay. Regarding the network pharmacology, what they ask, what they can do is there is a database called gene card. There is also database called string. Okay. Actually, 1 minute is, suppose if I share my screen. Okay, I think you people can see my screen, am I right?
Screen share.
Yeah, can you see the screen or no? Hello?
Yes, sir. Yes, sir. Yes, sir.
Yeah, this is where, madam. When you are talking about network pharmacology, actually this is the database that you can look at it, and from here you can look at this network pharmacology to select a target. Did I answer?
Yes, sir. Thank you. So we have one more question
Yeah,
from Dr. Ram.
yeah.
How the AI supports in pharmacovigilance department in future?
Of course. Okay. Even now also it is used. ChatGPT is effectively being used in a pharmacovigilance. Okay. Previously, the people used to write notes and everything. Now it's not necessary. Just go there. Whatever the doctor says, you record it. Whatever the patient says, you record it. Whatever the public say, you record it, and automatically you can put it. Okay. In that way, pharmacovigilance can occur. Of course, when there is a problem, the people can put it in a Reddit, the people can put it in a Facebook, the people can put it in e-social media, and
Yes.
ChatGPT can comprehend it. Okay. So pharmacovigilance means when you are collecting the data, now it is so easy. Actually, you can check all these social media tools, okay, to collect the information, and it is being used already. It's not in future. Even now also it can be done. Yeah.
Okay, sir. So we have one more question from Dr. Pradeep Bhavani. Is gene card is free?
Gene card is free. Yes.
Okay, sir. So a question from my side.
Mm.
So how we can shift towards a decision first approach, sir, where first identify the clinical or pharmaceutical decision and how we can improve that selecting that appropriate model.
Okay.
So once we fix to that question, how we can
Yeah.
identify that suitable model to
Oh,
further work.
that is experimental model. Am I right? You are asking me experimental model. Am I right?
Yes, sir. So how we can select that suitable model? How we have various types of models available. So how we can approach to that model?
No, madam, you are asking me experimental model in the laboratory model, or you are looking at
Using the
a.
database
Yeah,
and
okay. What you can do is you can ask. You can put all the experimental protocols. You can ask
yes,
ChatGPT. Ask what is good and what is not good in each and everything. It will compare, and you say that I want to test. This is my hypothesis. This is what I want. At the end, can you tell me which experimental model is good? It will tell you the experimental model. Hmm.
okay, sir. So any questions from the audience? Sir, small request from our audience, sir. Can you please share your PPT for our participants?
I'll convert into PDF and I will try to send it to. I'll do it, madam, later.
Can you share your PPT for our participants?
Yeah, yes, madam. Yes, madam. I will do it. I will do it. I will convert into PDF and I will send it.
Yeah, sure, sir. Thank you. So thank you, sir, for your insightful presentation. We truly appreciate the way you explain the concepts with the practical insights and made the topic very easy to understand. So thank you once again, sir, for your valuable time and expertise and excellent presentation, sir, on behalf of Mallikarjuna College. So I would like to request the principal, sir.
It was a very wonderful session. It has given information about the not only about Python, but the importance of AI. And Mallikarjuna said so many points. What AI cannot do it, cannot do the titration, cannot do the punching of a tablet and all like this. So many examples as quoted. So there is importance for the our human beings also and students and scientists also and AI. Only the how to incorporate the data. Based on that only AI is going to give the information. It's not completely AI is important. Our presence is also most important that he has specified. But as you all know that we must update ourselves. We should not be outdated. Sir has given, Mallikarjuna has given very important information, in depth, so many languages, so many softwares has informed us, and it will be very much useful for all the staff. The only the thing is how to utilize this opportunity. That's most important. All participants all over the globe, mainly from India, only of course maximum, and even my colleagues also here. So let them utilize this opportunity. Let them learn the things. That's most important. So that what is that you have studied, what is that you are teaching is of course it's also important, but at the same time, what is required for the present day competitive world? That's most important. Thank you, Mallikarjuna Garu. After a long time, we are seeing you again.
Thank you, sir. Thank
Such
you,
a
sir. Thanks.
very nice friend to me, very great man, man of knowledge, you can say that.
It's okay.
And I'm very fortunate that Sir was my colleague in the previous college, Vishnu College of Pharmacy, Bhimavaram, Mallikarjuna sir. And from there, some dispersion to different parts of the world. Of course, I'm here only. Sir has gone to various countries also. So very good development in my friend Mallikarjuna Rao. Thank you, sir. Thank you very much.
Thanks.
Right.
Okay.
Thanks for all the participants for your valuable participation. The next session, that is the afternoon session, will be starting by 2:30 p.m. We will soonly send the 2 o'clock. Sorry, it's 2 o'clock. We will send the link, so kindly join through the link. Thank you so much.